it's going to go. So this theme Babson, I'm going to divert from a usual hardcore scientific talk and spend a little bit of time on macro and microeconomics because I think this is going to be very pivotal in this field. I think what you're going to see in this field is what many of us refer to as technology leapfrogging, where technology that evolves and converges from multiple different markets comes together to make you skip over a particular part of the market evolution. And this has been known to happen in a variety of markets that are out there today. You can take the example of cell phones. People are no longer laying copper in certain countries, they're going straight to cell phones. This leaps over the entire 100-year of copper laying lines. And you can also see this in, perhaps more impressively, there's only 1 billion people in the world that are banked, and there's 6 billion that aren't. And digital currencies like M-Pesa, which runs in Kenya, there's 17 million people on this, responsible for about 70% of the transactions in Kenya, are now being run through digital currencies. They are going to leapfrog over making central banks fiat currency in war by doing this. This is going to be a tremendous number of people out of poverty, and we're not going to have 100 years of fighting over printed money. What we have also happening in some countries are solar-powered LEDs. We can get very efficient solar power now into nice battery systems that are coming out of places like Tesla, and of course, LED powers can skip over incandescents. So this happens, and it tends to happen when multiple technologies converge. And that convergence is in fact happening here in the United States, and many of you may ask, well, these all look like third world countries, why are you talking about this in the United States? We're not a third world country, are we? I'd argue our healthcare system is. In fact, the elderly generation is going to become a third world nation in this country because of some really daunting facts. The fact that the price of healthcare is going up, and it's over age, and there aren't going to be death panels that are going to milk you dry every last penny as you walk into the grave. This is the way the system is going to be set up to handle health as we age. Why is this happening? A lot of questions. It's a very controversial fact as to why is this happening. We have exponential price increasing in drugs. And in 1962, the FDA made some changes here, had a little bit more power to its blood money, and the new chemical entities coming out of the FDA are plummeting. This is not going to help this problem. So why is this happening? There are many well-intended people at the FDA, and there are many of them. They're brilliant scientists. They get into the field because they love medicine. And despite this road being paved with the best intentions, we end up with the opposite effect. And I'm going to argue the reason why is because of macroeconomics. It's because the FDA is a monopoly. There's 330 million people that are forced to work with it. When you vote, you can't change what they do. They don't have any competition. This means their prices go up over time. This is a standard understanding in economics. Competition drives creativity. It also creates pricing signals. And pricing signals are very important for market evolution. When government gets in control of insurance and they start mandating that people buy a product, do you think that product's going to get better if you're forcing them to buy it? Do you think the price is going to go up or down? Do you think it's going to get better or worse? Most people will argue it's going to go in the wrong direction. So there's a great book out there by John Goodman I encourage anyone to read called Priceless. It's the study of the fact that in the healthcare system, we don't have prices. You have copays. The insurance people will obscure what a procedure actually costs by paying half of it, socializing some of the people who can't pay it into other people who can. It's impossible for an entrepreneur to know what to fix. What has a really good market? If I make this technology, am I going to actually become profitable? You don't know if you don't have prices. So let's take a look at what this has done. Many people have noted that all of these illegal drugs don't kill nearly as many people as the legal ones. Thank you, FDA. Okay. This, since 1999, has been getting worse and worse and worse. So when people are talking about scheduling this and putting this under their purview, we have to ask, is that really the right thing to do? The black market has done a better job. The black market drugs kill less people. Now, if you look at what one of the causes of this, you can go back to 1992 and look at the PDUFA Act. Okay, the PDUFA Act allows the FDA, who already gets money from the tax base without really much congressional approval, to go and collect fees from drug manufacturers. Okay, so in any other business, this is called a bribe. Okay, you have to really think about that, let that sit in. The people who are regulating our drugs are on the payroll of the pharmaceutical companies. Is it any surprise that we have an increasing rate of pharmaceutical fatalities? It's not. This is probably baked right into the macroeconomic equation of what we're doing. So how do we leapfrog this? Because this is going to kill us. The way we leapfrog this, we believe, is that you take newer technologies that are coming out for personalized medicine, And we start treating each individual as an individual as opposed to a member of a herd. And when you start doing that, medicine makes a lot more sense. So back in 2003, a company that we were involved in up in Beverly, Massachusetts, 19 people built a DNA sequencer that was 100,000 times faster than what was conventional use. This didn't come to market alone. It came to market with another company called Celexa, Helicos, Complete Genomics, and Roche, 454. All of these technologies combined to drive a hyper-competitive market that dropped the price of sequencing 100,000-fold in a few years. This is Moore's Law. Moore's Law is famously coined by Gordon Moore, who is the founder of Intel, and he noted that every 18 months the computer processing speed doubles. That's an incredible metronome to rate your business. Are you keeping up with Moore's Law or aren't you? Well, the sequencing industry is. And so where this helps is it helps to bring in this concept of being able to sequence an entire person's genome for the cost of an MRI. Now, doctors don't have to guess. The other thing that we think is going to converge with this, and is going to help propel this leapfrogging event, is an over-the-counter open-source pharmacy. If you look at the pharmaceutical repertoire of cannabis, it is larger than Merck and GlaxoSmithKline, and it's all about become over-the-counter and open source. That's an incredible opportunity to merge with personalized medicine, because then we can actually put it to use. So we set out in 2011 to start doing this. We sequenced the ChemDog genome back in 2011, and we put it up on the web for free. And we did this for a reason, because if you look at trying to sequence actual human genomes, which we do at Courtagen, there's 20% of the human genome is patented. So if you want to sequence a kid with epilepsy, you've got to navigate. There's something like 4,000 patents holders in the human genome. Now, the number one patent holder of genes in the top 10, I should say, not the number one, in the top 10 holders of genes in the human genome program. Anyone know who one of the top 10 is? It's very similar to who owns the Hampson patent here in CBD. It's the NIH. So we as taxpayers paid billions of dollars to sequence the human genome. The NIH has been patented, so if we want to go back and sequence it, we've got to charge people more because we've got to navigate the government patents. That shouldn't happen. And we're not going to let that happen on campus. So we put this public on the cloud then to enable an open source genome. We are going to try and build some technologies that are proprietary to help keep this safe, and we're going to touch on that in a little bit. The other side of the business that we think is really critical to this is sequencing the patients to understand which one should get which cannabinoids. There's 80 of them, and you can't just throw them all at them. There is an entourage effect, and that's important, but it'd be nice to have a little science wrapped around how to fit the right drug for the right patient. So we've sequenced over 6,000 patients at Courtagen as a clinical test. We have a CLIA and CAP-approved laboratory that sequences these things for clinical, not clinical research, but for clinical tests, insurance-based. And about 500 pediatric neurologists that order these tests to sequence kids with epilepsy, to sequence kids with autism, mitochondrial disease, and developmental delay. And the interesting case in many of these diseases, like epilepsy, it's clearly endocannabinoid driven. If you have a sodium channel mutation, we know that you're probably going to respond very well to cannabidiol. But interestingly enough, not everyone responds perfectly. There's perhaps a way to stratify that. So we think these two approaches are critical to working together. But the important thing to note is that the FDA model of approving drugs is to treat everyone as a herd and take averages and see how many fall out. Many drugs drop out of the pipeline because this herd mentality is not effective anymore. Okay, we have a few of the drugs that are blockbuster drugs, like aspirin. Those aren't. There are no more of those left. So what we have to do is sort the patients out based on which ones have drug-drug interactions and certain genetics that might predispose them to perhaps not be able to metabolize CBD. There are two liver enzymes in your liver that cannabidiol shuts off. It potently inhibits CYP2C19 and CYP3A4. Those are the very liver enzymes that are required to metabolize most other pharmaceutical drugs, particularly a lot of the anti-epileptic drugs. So when you put a kid on CBD, if they're on 3 other drugs as they are with epilepsy, you can run into some problems. You have to be careful with this. So the ideal approach here is to find out which one of these cannabinoids appropriately go to which one of these patients, and we think that's important for us to get some science wrapped around the plant and some science wrapped around the patients. So here's a case of, we sequenced 960 mitochondrial patients that were referred to us from physicians. Okay, 73 of them had chronic pain, and they all happen to have a variant in the ATPase domain of this gene called TRAP1. It's involved in cancer. We don't really know much else about that gene. When there are mutations in this particular domain down here, this ATPase domain, if it's at a particular amino acid, one like right here, you end up with 3 symptoms: chronic pain, chronic fatigue, and dysmotility. These variants exist in about 1 to 2% of the population. The reason this is relevant is that most of these patients are seeing, I shouldn't say most, there's only 16 in this category, but a few of them are in fact seeing a good response with N-acetylcysteine. N-acetylcysteine is a strong antioxidant. You can get it as a supplement. Cannabidiol is better antioxidant. In fact, it's easier to dose. It lasts in your body longer. And we believe this would probably work even better, although I think patients are probably too afraid to try, just due to the nature that one you can get from Whole Foods and one you can't. So what's important about this is, for the longest time, the medical marijuana movement or legalization has not really responded well to the concept of pain. You have pain, sure you do. Your arm's broken, you need weed, I get it. This has been considered too subjective of a criteria for physicians to be able to hand out pain meds. A lot of trouble with opiates, as people mentioned earlier. Well, if you actually have a sequence, you have now objectified what used to be a subjective problem. These patients actually have a damaged enzyme, and we know that they actually need some type of medication, and we can begin to change this dialogue. This dialogue is no longer about subjectivity. We think that's going to happen in many cases. It's going to happen in epilepsy, it's going to happen in autism. We're going to stratify these diseases into subcategories genetically and know which ones need certain therapy. So, everyone frequently asks, why do you obsess about cannabis safety? No one's ever died, and that's partially correct. However, it's usually thought about cannabis in terms of recreational use. When you start getting involved with patients, some patients have symptoms that when you add cannabinoids to them, you can harm them. You've got to be careful. There's some cases in the literature, actually, of documented fatalities, not from cannabis, but from the mold that grows on it. So, improperly prepared cannabis, one would argue. There's probably only 5 of these in the literature of aspergillosis, which is an aspergillus bug that grows. It can make aflatoxin. If you really had to squint and try and pull out more cases that were questionable, you might find that in the GW trial, 20% of the kids got worse than Epidiolex. It's not really spoken about because 50% got better, and it is a miracle drug. But why do those kids get worse? When they get worse, they take them off that drug because seizures can kill you. So you got to be careful thinking about this as 100% safe. It's really dependent on the medical context. Now, this is a very controversial one, and Lester, I wish were here to debate me with this, because he is correct that schizophrenia has not been increasing despite medical marijuana use has. So it generally does not cause schizophrenia. However, there are some very, very rare SNPs in the cannabinoid 1 receptor that they have shown have higher correlation with people getting induced schizophrenia. But there's such a small minority that they're in the noise of what we can measure in the general population. This is probably why the schizophrenia debate continues to this day. And then we've seen a couple cases of people on high doses of edibles, and they lose judgment and do foolish things. There's one in Denver, I think, that apparently jumped out of a hotel room, and that's been sloshed around the industry. And there's been a couple cases of fatal tachycardia attributed to cannabis use in London. But we don't really know if it's cannabis or if the patient genetically had a long QT syndrome. There are such things as like Len bias, right? So those genetics are known. You need to sequence about 200 genes to really understand that genetic picture. And of course, there's cannabinoid hyperemesis syndrome. Many people don't know about this, but it's in fact an allergy that some people have to cannabis, and they go into cyclic vomiting if they have too much. Usually chronic use and very rare. So what can we do to bring safety into the field? Despite the fact that this is the safest plant, we think, in the world, there is still a fear-mongering effect that's in the field. Okay, so we want to try and bring the best technology to bear here. The first 3 product lines that we started on was, first, we want to be able to share the genetics of this plant. DNA, unlike material from the plant, DNA can go across state lines. DNA from the plant, as long as it's purified from the stalk or leaf. If it's purified from the flower, it can. Same DNA, don't ask. It makes no sense at all. So we built a DNA purification kit that uses these magnetic particles. A lot of this technology actually came out of the Human Genome Project that we've worked on. And this is just used to be able to purify DNA very quickly so people can share some of the genetic information. And then the other kit that we're exploring right now, it's now in a couple laboratories across the country, and it's rapidly getting installed, is a quantitative PCR system to pick up mold and fungus and other bacteria like Salmonella and E. coli that people worry about. Now, despite the fact that the presence of this type of thing in cannabis is extraordinarily rare, it is kind of mandated that you do this in many states, and it's mandated actually into a lot of the food testing industry as well. This did not come out of regulation from the DPH per se. There was a testing company in California called Seedfield Labs, and there is no mandated testing in California, and they decided to do this voluntarily, and the market responded pleasantly with support. So it's an interesting lesson on regulation there, that you can get market-based regulation. Many of the states are now emulating what Seedfield Labs did initially and putting that into print. And then finally, we want to play around with some cannabis genetics because we believe you can type for the sex of the plant and the THC potential and CBD potential genetically. And that's become very valuable for work for some of the growers. Okay, so why do we want to use quantitative PCR? Culturing bugs to figure out if they're there takes a long time. It takes 48 hours usually, and you have to grow this on a petri dish or in culture, and only 1% of the microbes can actually be cultured to this day. Technology's been around for 100 years, but that hasn't changed that fact. It's also, as I mentioned, very, very slow, but the other challenge is it's not very specific. When you try to brew a beer to detect whether salmonella is present there, many other microbes can grow in a salmonella broth. It's not really specific. You can get other things growing that you think are salmonella, but they're not, and using a DNA fingerprint is far more specific, which is why we like sticking with PCR. The culture can be easily counterfeited in many ways. It's very good to heat things, as we've heard, to decarboxylate and maybe sterilize, but the heat kill doesn't get rid of the fungal toxins that grow. So aflatoxin is one that is made from aspergillus, and you can certainly heat kill it so you won't get a lung infection, but the aflatoxin can still be there in very small amounts. Aflatoxin is a carcinogen. The only reason why the physicians worry about this is that it requires the same liver enzyme I mentioned earlier for clearance in the human body. So if you're on CBD, you're not going to clear aflatoxin. It's sitting around. And the main issue that we find from an engineering standpoint is that the culture-based systems, basically, when you get no signal, it equals safety. And that's an engineering flaw in any safety system, right? Okay, I got no signal, it means it's safe, right? You always need a signal. And this is why we build in PCR, an internal control. And this internal control actually took us down an interesting path of sequencing dozens of cannabis genomes. So this marker up here on the quantitative PCR, what you're looking at here is time. This is roughly minutes. This is roughly hours using a culture-based system for limits. And you're looking for signals, in this case, it's CO2 that's building out that we're measuring, and the indicator size changes color. Like in this case, you're measuring a fluorescence molecule in the DNA that's building out over time. But we're measuring two different things. Here's the yeast and old DNA sequencing like that with time as we watch your blockings that came in 20 minutes instead of a 5. This here's a single copies cannabis gene. And we actually get to know examples of things about this from tomatoes, amazing, pressure. So gene is we've had sequences of substances which is HGH, which is a very nice genomics going on in this region. We have temporal or genetics production of hyper variable regions because everything's free after that. So we had a gene, and that's my ever reactive test. Yes, present black correctly, and this works. And how much genes is there as the rest of the life. And that's how this test works. Provides information is not all present. And just a side note, here's a sample that we'll see visibly actually on culture. This is the problem with culture-based systems is that not everything grows in the rods. PCR the other issue that you have with whatever you're doing in microbiology and cannabis space settings laboratories is that you actually have to amplify the contaminants. It's kind of dangerous because you're trying to grow cells in order to detect it. So it's very important that laboratories have clean standards and microbiology techniques. And what we put into place in this assay, we published in 2 different journals that builds in a decontamination system that we also think gets around a lot of gene pads that are out there. The way we do this is we typically amplify DNA. You have 4 letters in there when you copy DNA. We've added a 5th, but an enzyme targets and destroys. So every time we do a test, it self-destroys itself. So the DNA can't be around to contaminate someone's future testing of their sample. So we think that's actually critical to get this out into the field and to make it really bomb-proof. Now, there's another aspect that growers have asked us about, which is, well, these pesticides are a real pain to detect. The pesticide testing is probably the hardest testing that can be done out there. Can we establish a growth environment this legal? How do we monitor the health of an entire grow? Well, in the design of this complicated qPCR assay, it's quantitative PCR, we did something that enables this amplification reaction to actually then be DNA sequenced. And this is handy if you want to do any type of microbiome work on the plant. And this is actually built into the yeast and mold test that we run, is that we can, if you get a positive result, you can then go and sequence to find out exactly what type of bug it is. So you don't get that if you use a petri dish. You get a little colony on there, but you don't know what organism it is. You have to resort to some kind of sequencing approach to do this. And when you do this, you get a really nice spectrum of all the different bacteria that are living on the plant. So we've got lots of penicillium, which is nothing to worry about. That's actually an endophyte and helps the plant grow. It's very healthy. Magnaporth is a blight. This is probably not good for the plant in terms of its health. And aspergillus is something we worry about for human health, because aspergillus is something that can actually make aflatoxin. And then, of course, if you get down here, there's fusarium as well. There's, a whole whole list of these pathogens for plants that people want to keep their eye out. But what folks are envisioning doing with this is periodically sampling a large grow like this, sequencing its microbiome, and having a surveillance system that tells you that, okay, this sector of the growth room is getting a lot of detritus. Let's cut it out. Let's remove it before it spreads. That way we don't have to go and spread pesticides everywhere and induce excessive testing later. And you can go through all the different microbiomes we've done, and there's only a few areas where you get spikes of differential microbes growing in different plants. And they tend to be in the Penicillium area, the Aspergillus, and this one here is probably a human contamination from dander. So hair nets are probably advised on that part of the grow room. And you can compare these one to another and do all types of interesting work as to how they differ from ChemDog to Lester Grinspoon, which is, yes, the name of the strain, not a person. We didn't sequence him, we sequenced a plant named after him. And Granddaddy Purple. So this we think is going to help eliminate an excessive use of pesticides. Probably not completely, but it may also give us some understanding of why does the plant grow certain cannabinoids. It evolved growing these things to actually manage its microbiome. It's thought to be a pesticide and an insecticide. So having this information is going to be very important, I think, in understanding why certain genetics express certain cannabinoids. Now, the other thing we can do from a hole punch for leaf now is go and figure out if they're male or female. And the reason we care if they're male or female, many of these large grows will in fact clone things, but when they want to find new genetics, they cross, they seed things, and then they have to weed half the males out. Because the males, they stick around, they pollinate the entire grow, and they destroy your crop. They seed up, and you lose cannabinoid content. There was a gentleman who did some amazing work. Mandolino did some work many years ago before we even had a reference genome to figure out what bands in a particular PCR assay would tell you whether it was male or female. You can tell it's kind of squinty and hard to read, not very digitized. And so we went to aim to convert this into a quantitative PCR assay. And looking at this region, all the genomes we've sequenced, this region has actually got 500 copies of itself in the genome. It's probably not a good marker for doing this as we expand into more genetics. And so we've remodified this assay so it's not sitting in something as complicated by going through and sequencing a bunch of male genomes. Imagine if you take a bunch of male genomes and sequence them, take all that data and scan it across to females, and the data that sticks to females is probably all female and X chromosome. The stuff that's left over is the Y chromosome. And we assemble that Y chromosome and it tells us what areas to target so that we can design assays that say, okay, there's a Y chromosome here, or there isn't. And this is a very quick, basically, 60-minute assay that tells you male, female off of the leaf punch. We can do the same thing with THC and CBD. These are also genetically encoded. Now, we're not going to get to the precision that Scott's going to get by measuring out the whole plant because the genomics is really the deck that you're dealt. It's not how you play it. And many growers can actually take certain genetics and get them to express slightly differently than others. And so the real magnitude of THC and CBD is probably more growth technique. But its genetic destiny is known to be tied to a couple of DNA markers that we can also convert into quantitative PCR tests. So Mandolino, once again, published this great paper that showed that plants tend to make all THC, all CBD, or something right in the middle. Not too different from what Scott showed, right? It's very rare that you get them sitting off in these axes. And that is because of a particular region of the plant that governs which way the plant makes THC or CBD. So it all starts with this precursor, and the enzymes that make this are competing for that precursor. So it either goes this way, or down the middle. And that tends to be something that is very important to growers when they are ripping plants out of the ground. They want to save the males with high CBD right now because CBD is in high demand. There's also been some variants that we found when we sequenced the genomes that imply there isn't one THC synthase in a given genome. There's sometimes 2 or 3. The plant can replicate this pathway. And some variants that they found over at GW Pharma, that was just published a few weeks ago, imply and reproduce this, that genotyping some of the regions in the THC and the CBD synthases will help understand the magnitude that you can in fact make. Whether something is a 15 or 20% THC strain is probably more related to some of these variants than the last ones. So we're working on converting those assays that can go onto a quantitative PCR station, and growers can then check for this and help segregate the plants. So the final thing that I want to touch on is ending the strain name game. Many of you may be aware there's no trademark system in cannabis since the federal government won't accept any trademarks, so people tend to rename things to whatever gets the highest value. There's counterfeiting going on left and right. I wouldn't be surprised if there's 10 versions of Charlotte Web that are actually out there that have nothing to do with the original genetics. Morally Naturals is probably going to capture twice the price of anything else. All right, so how do we end this? Everyone's been asking to be able to sort this problem out. And one way you can do this we believe, is by doing a process called RADseq. Now, to sequence an entire cannabis genome, it's probably a couple $1,000s today. That's too expensive. We want to get that down over the magnitude. So what you do instead is you find a way to sequence the first chapter, the first page in every chapter of the genome. And then you compare those first chapters genome to genome, and you can find really interesting phylogenetic relationships this way. So we developed a technique that does this, that sequences, after we do a restriction digest of the genome, we take a little slice of it on this machine up here, and we sequence a couple million reads in, and we get ourselves 100,000 SNPs, single nucleotide changes of the plant, and we use that to build phylogenetic trees. So we get these nice little pileups in certain regions of the genome. This is the first page of every chapter that we sequence. And then we get, we measure where these differences are, and we use those as markers to track different phenotypes that people chemotypes. Some people aren't just looking for THC and CBD, they're also looking for high diversity, high aridity, different terpenes. These things are actually all pharmaceutically active. In fact, beta-caryophyllene is one that is got tons of clinical literature on it, so it's a very interesting terpene from plants. Not unique to cannabis, but it's certainly important when it's thrown into the mix. Okay, so how do we then solve this problem? This is really a community problem. When we sequenced ChemDog, everyone said no you did, because how do you know it was really ChemDog? You know, that's folklore. So we believe this is what is known as a distributed consensus problem. You have to get the entire community to agree on what's what. How do you do that? Well, the Bitcoin blockchain system does this. We can just steal that. Effectively, this is a distributed consensus system. If people submit genetics to a testing lab and you get these tests done, you can effectively be building a Wikipedia that's public. And every time someone sees Blue Dream enter into this, the next 10 Blue Dreams show up and have a different story, the phylogenetic tree shifts. And this can all get done with blockchains. It's a very interesting approach to potentially solve this in a Wikipedia model. We don't believe this belongs in any one company database, although we're conflicted that we want to sell you genetic tests to do it. The database itself, we think, will only stick if it's a Wikipedia model, because this industry, I think, has a general distrust of this thing getting wrapped up into something like Monsanto or what have you. So that's the only way we see this naming game ending, and we're starting to do this now. So we had a strain come to us from Mark Emery out of the workshop in Seattle. They had found a strain that came out of Australia. There's a lot of folklore over this strain, saying it was some fourth species of cannabis. We didn't believe it. Somebody crossed this with a sativa, and we got that cross out of Seattle, and we ended up sequencing it. And interestingly enough, if you take all the reads from this genome, there's a bunch of other genomes we sequenced, and if you map them against one of the references, you'll get about 99% of those reads mapping to reference. Hemp, however, gets about 97.7% mapping to that reference. If you were to map the human, the bonobo reads, an ape, to a human, you'd have about 95% mapping. And this Australian cannabis strain is even further from that. I think that's what's so fascinating about the genetics of this plant, is that you have sativa, indica, rhodorellus, and maybe this thing that can still interbreed successfully. And you can't get that with bonobo to human. So it's a very dynamic plant. Now, this is the phylogenetic tree of it. The next nearest neighbor is hops. So we took 4,000 SNPs in this case of all these different strains that we've sequenced. You'll see one in here called Lester Grinspoon. I got cut off here. That's not a person, that's a strain. And this Australian bastard is sitting out here away from all of these different indicas and sativas, closer to hemp and near hops. Now, this is probably because this was crossed with a sativa, not with the hemp strain. If it crossed with the hemp strain, it'd probably be sitting way out here. So it's quite possible this is a fourth cannabis species. We don't know how useful it is. It doesn't make a lot of terpenes or a lot of cannabinoids, but it's drought resistant, and I think some people in California are trying to get this into their gene pool. Who knows? But anyway, I think the take-home lesson here is that personalized medicine is here today. I encourage everyone to get sequenced, because this is the most valuable thing I think you can do to better understand your health. But that market-based regulation is possible. It's actually better. And we see this now on eBay, Airbnb, Amazon, Uber, all of these mechanisms on the internet for us to communicate and return information about what's safe and what isn't safe can readily run in this industry. After all, it's the safest drug that we've seen. Why wouldn't this model work? We believe that's what's probably going to happen as this goes to the dispensary model, and it's probably not well suited for an FDA $1 billion trial. So with that, I will leave the questions. There's some folks in the audience. I point out the folks from New England Biolabs here. Joanna Bybich introduced me to Lester. That's how we got some of this stuff sequenced. And there's a handful of folks at Courtagen that were working really hard on this. And we even have some support from a reggae band called Slightly Stupid. So they've given us rights to some of their music to help push iPad apps. So we like thanking them. Questions?
I have a question about the London cases.
Yes.
I was reading that they were actually using synthetic marijuana. Is that?
That I wouldn't be surprised at. I rule all synthetic stuff out because that stuff is not cannabinoids.
No.
Somehow it's labeled as cannabinoids because if you trigate it, it sticks to C1 receptors, but it hits 1,000 other things. It's those things look like fluorophores to me. I wouldn't go near them. So whenever I see something written up as a synthetic cannabinoid, I know it's a shill. It's just a smear campaign against the field. I wouldn't pay any attention to it. There have been 2 cases that were deemed cardiac from THC, and there is some evidence that THC can induce tachycardia. That's probably not good if someone has long QT. And so the argument in those papers, they did do some genetic sequencing of those patients to see if they had any mutations, but they only surveyed a couple genes. And to do that completely, you have to go through about 200 genes that are known to be involved in that problem, and the paper didn't address that. So we've tried to reach out to the authors and ask them to collect some of those patients to see if there was something amiss, but there hasn't been much response in the ability to get DNA back from those relatives. So it's still a bit of a purgatory case.
Great information, thank you very much. And how do you get sequenced?
How do you get sequenced? So I'm glad you asked that question because it's illegal for us to directly sequence you. The FDA has once again come in to save the day and has deemed your genome is too dangerous for you to know. So you have to get a doctor to write it. They can write a prescription to get you sequenced, and there's a variety of clinical laboratories that will do this. They probably won't do it unless you have a medical condition. And even if you do have a medical condition, they may choose one lab over another based on their specialties. We tend to focus mostly in neurology because we feel that has the biggest impact with the endocannabinoid system right now. We haven't been doing any work in cancer. But it's pertinent for anyone who does face cancer. That's probably the best thing you can do is sequence your genome and the genome of the tumor because that showcases what's going on.
And with your personalized medicine, you're anticipating that someday down the road people may be doing this for their own preventative?
They already are to some extent through a company called 23andMe. Now, the FDA shut them down because they were going direct to consumer. They've recently allowed one of the 500,000 variants on their list to go direct to consumer. They expect one by one, every 2 years, one of those 500,000 will make it to the FDA. So I don't know how that's going to break down. The good thing about genetic information is you don't have to be confined to the United States. Information of your genome can flow in here legally, so you can get sequence of your data somewhere else.
If you get a prescription from your doctor, you're kind of care family doctor, and eat this.
No, if you get it, it has to be ordered from a physician. But and we won't sequence cases just for sort of recreational discovery. It has to be a CLIA CAP certified test. So you have to have like epilepsy, developmental delay, mitochondrial disease, or lysosomal storage disease, and physicians will send a test our way.
Do you get for children?
We're mostly children. This is the reason we do everything with saliva, is it's very difficult to get needles into seizing kids.
And is it covered by insurance?
Well, that's a tricky question. Sometimes, sometimes not, and it's very much dependent on, I think you get a kick out of going to a lab. We have 3 people run the whole lab, and does 400 patients a month, and we probably have 20 people in the back office with typewriters dealing with insurance companies. So it's very much, about 50% of the time it gets reimbursed. Is generally what it is, and it very much depends on the plan that you have. Now, my general fear of insurance plans in general, what's going to happen as they've been given a hall pass on having everyone forced to order their product, is that the minimums are going to go up, so your yearly deductible is probably going to creep up over time, and your copay is going to go up. And there's not much we're going to do about that. So the ability to be able to get a sequence and whether that's actually in the plan that you have is likely to change. I think the one ray of hope here is that they are seeing that 40% of the time when you do genetic sequencing, you pinpoint the nature of the disease, and you short-circuit this whole diagnostic odyssey that costs them a fortune. So there's a high financial motive for these insurance companies to eventually get on board. It's just going to take them, I think, a couple of years to recognize those returns are real. And in which case, I think it will be built in, probably 3 to 5 years will be built into many more plans.
Excellent. I'm great to keep up with you because it was so correct. I think we'll be glad to know the fact about 1980 we were actually advertising our maximum bought there in a community journal that is
Excellent.
Of aspergillus, and it actually bought and used by the medical.
There we go. Thank you. If you keep it too short,
related, say, 3 or 4 generations back, would you be able to identify the exact rate, or is it just a percentage that you have a likelihood?
So the question is, if there were 3 generations apart of plants, which DNA sequencing go to see that? I think yes, even today, with the cannabis references we have currently. The human
Right
genome
now,
has
as
got,
it stands, is it just a percentage of likelihood that
yes,
they be
yeah.
related within a
We'd
certain
be able to
amount
draw
of
a
generations?
phylogenetic tree, basically, off of 100,000 markers, so you have some pretty good precision out there. The only caveat for out there is the cannabis references that are out there today, some of the ones that we publish and others publish, are pretty rocky as genome sequences go. That's because the plant is so diverse. The mothers and fathers can vary one base and every 8. You and I vary 1 in 1,000. Okay, so the mothers and fathers are very diverse, and it's 65% AT, which means it's very hard to read because all the jigsaw puzzles look red, if you will. Right? The human genome is about 50-50 when it comes to AT content, so it's much easier to read. They're going to get better. There's new technology out that can improve these references, but I think you can still sort your problem out today. What's going to be out.
Do you find a difference in the genome sequence between a feminized seed and one that was pollinated?
That is a fascinating question, and I suspect that we've not done that, and I think you will. Because the feminization process looks like it's using silver nitrate, and we know silver nitrate actually sticks very potently to DNA. We actually used it in one of the DNA sequencers we built as a mechanism to cleave the sulfur bond in DNA that was special to us. And so it's fairly mutagenic. So I think that process is doing something with the MD genome to force these things into making only male or only female based seeds. And I don't think anyone knows, but it's a fascinating question. Oh, I guess.
Yeah, excellent. I really enjoyed this very much. You know, one of the questions I've been looking at is that how certain illnesses come from certain areas. When I was out in Washington State, I saw some of the most rarest diseases, and I did not know it was because of the plutonium contamination that led to it. And my question always is, do we need to grow cannabis in certain areas to treat certain illnesses? Because, you know, we talk about attenuators turning on and off genes.
Right.
Is that related to trying to grow the appropriate cannabis for the
I
appropriate
think
illness?
I would bet, I'm not a grower, so I don't know the answer to this, but I would say if you talk to anyone in the wine industry, they'd say absolutely. Soil, pH, and microenvironments, and the microbes that are in that environment make a tremendous impact on whether a clone does X or Y. And I think it's very important. It's an important statement to put in context with genetics. Because genetics, as mentioned, is just the cards that you dealt. It's not how the environment plays with them. So I think that's going to be the case, that you're going to find some environments, different light cycles, different soils that are going to produce, maximize certain terpenes and certain chemical expressions. There is a really fascinating theory out there that I tend to gravitate toward from Sinclair on this, where he believes, the hypothesis being that the plant produces these chemicals as a trigger to signal to the people or the organisms that are fostering the plant. It creates these secondary metabolites so that your body actually responds differently and begins to prepare for starvation. The plants are making these chemicals as a mechanism to communicate to those that are farming it that change is on the way and something's wrong as the balance changes. So if Sinclair is correct, then absolutely.
Thank you.